Oh, come on. Thing. So I had the microphone off, so that's why. So now it's recording. Let me share my screen. All right. Are there any questions before we begin? Today is October 19th. And let me think about that. We're supposed to be starting chapter 4, which we did. That's right. So we should be starting chapter 4 and getting about halfway through it. There is no lab for today. Is that correct? Did we do lab 5 on Tuesday? Let me look into that.
Yeah, we went over it.
Okay, so there is no lab today. Thank you. I will be logging in for the first 15 minutes of lab time to answer any questions. And if there's more questions and lots of students logging in to ask questions, I'll be longer than that. But if nobody comes in, I will log off after 15 minutes. So if you want to talk to me or you want to ask questions, make sure you're there from 6:30 to 6:45. Because after 6:45, if there's no students asking questions, I'm going to log off. Let me remind you, you should read the unknown project instructions file on Canvas. I will discuss that in our next lab. And we'll actually start the unknown project file next week. Any question about any of that? All right. I thought I had the lab up. It looks like I don't. All right, so we had talked about prokaryotic cell arrangements, also called cellular growth arrangements, and that's when the cells stick together, what arrangement they form if they stick together. Now, remember when you're talking about prokaryotic cell arrangements, that one of the possible arrangements is singlets. You can have a single cell. The students, for some reason, tend to forget that. And then the other thing to remember is in the cellular growth arrangements, you never have one cellular growth arrangements if there is any arrangement over a singlet. Obviously, you can have only singlets, and the spirillums and the spirochetes only exist as singlets. There may be a few other species that also only exist as singlets, meaning they just don't stick together. But with the cocci and the bacilli, you can have other cellular growth arrangements and they can break apart. Let's go to the cocci. Like the streptococci can break into diplococci, and then the diplococci can break into singlets. So with the streptococci, you will see in the slide streptococci, diplococci, and singlets. With staphylococci, you might see all of the cell arrangements because the staphylococci can break into Sarcsinia, which can break into tetrads, which can break into diplococci, which can break into singlets. And then if the staphylococci breaks a certain way, you can actually get streptococci. What you do when you have a mixture is you look for the highest order of cellular arrangement, and then you call it that, meaning like right there, there's a singlets, and this right here would be called Staphylococci because we go with the biggest order, not the smallest. And that one here, the Diplococci, that actually looks like maybe it's just two. All right, any questions about cellular growth arrangements? If not, let's talk about the structure of typical prokaryotic cells. You can see many of the different structures shown in this slide here that prokaryotes have. There are only 3, possibly 4, structures that all cells have, and that is a plasma membrane, cytoplasm, and then DNA. So all cells, including eukaryotes, have all 3 of these structures. You could also say that all cells have ribosomes, but the ribosomes of prokaryotes are different from the ribosomes of eukaryotes. So sometimes the ribosomes aren't counted. Other cell structures include a pilus, a sexual pilus. We'll talk about that when we talk about genetics. Only some cells have a pilus. A capsule, that is a secretion made by a cell, and the secretion is external to the cell. Not all cells have a capsule. In fact, of these 2, actually a minority of cells have a pilus or a capsule. Most prokaryote cells have a cell wall, but not all of them. Certainly there's one family of bacteria, oh gosh, I just dropped the name of that family. Mycoplasma, that do not have cell walls. And then, as you know, not all cells have cell wall. All animal cells lack a cell wall, and many protists also lack a cell wall. Plasmids, many bacteria have plasmids, but not all bacteria. And there are some yeast that have plasmids too. And yeast, if you don't know, is a eukaryote. Flagella, this is the structure that prokaryote cells have, meaning bacteria, since that's what we're talking about, that give the cell locomotion. Eukaryotes can move by flagella, but they can also move by cilia or pseudopods, and we'll talk about those a little later. Have I talked about? Yeah, I already talked about a capsule, a cell wall, plasma membrane. Oh, and some cells, I'm not sure if this is a majority or a minority, I just don't know. But of bacteria, some of them have fimbriae. The fimbriae are shorter than flagella, as well as the pilus. And they are mostly used by attaching the cell to something. So the fimbriae attach to something. Any question about any of that? All right, the glycocalyx, which we did talk about previous when I was talking about the capsule. The glycocalyx is a secretion by some prokaryotes. It's not most of them, but there are a number of prokaryotes that have a glycocalyx. It's outside the cell wall. It's usually sticky, and there are 3 types of glycocalyxes, and you can pronounce that glycocalyx, or you can pronounce that glycocalyx. Both pronunciations are correct. There are 3 types. The capsule is a neatly organized glycocalyx that has structure to it in many molecules, meaning that it has many other molecules other than water. And if you can see it under the light microscope, it is a capsule. The capsule is important because it can prevent phagocytosis, meaning a white blood cell cannot phagocytize a bacteria that has a capsule. A second type of a glycocalyx or calyx is the slime layer, and it is unorganized. It's very loose. It's mostly water. You cannot see the slime layer with a light microscope. To see the slime layer, you have to see it with an electron microscope. And that's mainly because it's just mostly water. Any question about that? All right. And then there's a third type of glycocalyx. We won't talk much about it. It's the extracellular polysaccharide, abbreviated EPS. The extracellular polysaccharide is in a biofilm. What happens is there are different bacteria, which will be in an area. So we can say this cell here, and then we have another cell here and a cell there, and they can secrete a glycocalyx, and then that glycocalyx eventually becomes the biofilm. So that'll be a biofilm in this whole region. And there's one biofilm which includes this cell and that cell and that cell and all the other cells in a biofilm. The cells are usually stacked on top of each other and then grow within the biofilm. Anyways, the extracellular polysaccharide is a glycocalyx of cells in a biofilm. Any question about any of that? The main one we will talk about is a capsule because you can see it under the microscope. And the capsule also gives, let me see if I talk about that in the next slide. No, I don't. The capsule also gives important properties to the cell, and that is the immune system has a hard time fighting off a cell in a capsule because there's no phagocytosis of the bacteria in a capsule. Actually, I shouldn't say there's none, but it's reduced, greatly reduced. And that is actually one reason why Streptococcus pyogenes, when somebody gets a sore throat from Streptococcus pyogenes called strep throat, why the patient cannot typically throw off that disease. And then the strep throat just lasts and eventually gets worse. And in the old days, before antibiotics, strep throat would become a very serious disease because it just tends to get worse and would infect the body, causing heart disease, causing, I think it was rheumatoid fever and what was that other one called? Sorry, scarlet fever, a bunch of other diseases, which are very rare nowadays, because when somebody gets a sore throat that lasts a long time, they go in and see the doctor, and the doctor tests them for strep throat. And if they're on strep throat, you're giving antibiotics, and then that'll take care of it. And so oftentimes, at least nowadays in the United States and first world countries, somebody who has strep throat, the disease never goes further than that. Has anyone ever heard of scarlet fever? I think the other one is rheumatoid fever. These diseases were very common before the introduction of antibiotics. And it was because the body had a hard time throwing off strep throat. Has anyone ever had strep throat? I have.
Yes, I have.
Yeah, I had it 5 or 6 times after I first got it. And the doctor did warn me, I think when I was in 7th grade, he said, and you might get it back. So if you get a sore throat, come to me again. And I had it like 5 or 6 times up until I was in college. Fortunately, when I was in college, I never got strep throat again. And the reason was the antibiotics dropped the numbers to very low numbers. So I no longer had strep throat, but I didn't fully get rid of the Streptococcus pyogenes. And every once in a while, it came back on me. And it's because it has a capsule. So the immune system has a hard time. Where's my mouse? Immune system has a hard time removing it from the body. What's your story on strep throat? If you care to state. If you don't want to state, you don't have to.
Oh, nothing. I've just had it a couple times, just and then given antibiotics. My kids have had it too.
Oh, geez, yeah, it spread to your kids. Yay. Once you have strep throat, you have to be careful because it is easy to spread. But you had it repeatedly, so that means it came back after it was cured for a while, but you still had it at low numbers, and so it came back. All right, flagella is the structure that prokaryotes have. We're talking about bacteria, but also archaea. And that is if they move or they locomote, meaning they can swim in a liquid media, then they have flagella. If you have one, it's called a flagellum. But if you have more than one, it's called flagella. It's mostly outside the cell wall, as shown here. But if you look at it, there is a little bit that's actually inside the cell wall. And then it's also anchored on the plasma membrane. Flagella is made up of the protein flagellin, and you don't really need to know that, but that's just a protein that is found in the flagella. I don't think I asked any questions about the filament, the main part of the flagellum, and the hook, the part that attaches to the cell wall, and the basal body, the parts that's inside the cell wall and the cell membrane. I don't think I ask any question about that. I'm not going to quiz you on this. However, you need to know it for the lab. And that is the 4 basic arrangements that bacteria flagella can take is you can have monotrichous flagellum, because there's only one, and that means there's one flagella on one end of the bacteria. You can have amphitrichous, that means there's one flagellum on both ends of the bacteria. So there's 2, one on each end, and that's amphitrichous. And then there's lophotrichous, where you have multiple, we'll say multiple flagella on both ends, and that means 2 or more, but only on the ends. And then you can have peritrichous, where there are flagella all over the bacteria, and there's many flagella in the bacteria. Any question about that? Have I talked about flagella in the lab? I don't remember. Whenever you see a bacterial flagella arrangement, you will see more than one. Even if there's only monotrichous, you will see some bacteria without having a flagella. And that's because when the bacteria first is making the flagellum, obviously it doesn't have a flagellum, and then it makes it, and then it'll be monotrichous. If, on the other hand, it's an amphitrichous bacteria, it'll first start out having none, and then it'll make one, and then later it'll make one on each side, becoming amphitrichous. And the same with lophotrichous, and the same with peritrichous. So if we have a species which is peritrichous, initially the bacteria will have none, and then it'll make its first flagellum, and it'll be monotrichous, and then it'll put out more, and it'll be amphitrichous, and then lophotrichous, and then finally it'll put out most of them or all of them, and it'll be peritrichous. So in the species, which is peritrichous, you'll see some cells which are peritrichous, some which are lophotrichous, some which are amphitrichous, some which are monotrichous, and then some having none. Any questions about any of that? And like I said, you only need to know that for the lab. I won't be quizzing you on that. If cells are motile, that means the cells either rotate, having flagella, meaning they'll swim or run, or they can tumble. Maybe I can show it like this. Swimming is when the cell moves sort of like that, and tumble is where the cell tumbles like that. If cells move, they have flagella. I'm talking about bacteria cells. It's also true of archaebacteria. If they move, they have flagella. That's not necessarily the case if it's a eukaryote cell, and we'll talk a little bit about them later. Taxis is the movement toward or away from a stimuli. There are two types of taxis. There's chemotaxis and phototaxis. Chemotaxis is movement toward or away from a chemical, which is giving the stimuli. And then there's phototaxis, that's movement away or towards light. This bacteria is obviously not photosynthetic because the light source is up here and the bacteria are moving away from the light. So they are phototaxic, but in this case, they're moving away from the light. And whoever photographed this must have got a specific species to do that, because I don't know of any species that do that. All of the photosynthetic organisms will move towards the light. Any question about any of that? All right, let's talk a little bit more about axial filaments, also called endoflagella. I use the term endoflagella. It is a flagella, but it's an unusual one found in spirochetes. The flagella is anchored at one end of the cell, and then it wraps around the cell. Shown here, or this is an actual electron microscopic image where the flagella is shown wrapping around the cell. And it's hard to see, but that actually goes around the cell like that. And I'm not sure why we're seeing it there, probably because right there it's darker. We're probably seeing it through the cell. This endoflagella does cause the cell to move, but in an unusual movement. I'll get that going. Oops. Let me come back to my slide here. It looks like it's going to start. Nope, we got a commercial, so let's move on.
Switch to Chrome to protect yourself from malware, so you can surf without getting phished. Fast, safe, yours.
What's unusual about the
There's no place like
endoflagella,
Chrome. Get
Yes.
Chrome for Windows.
It's inside the cell wall. Let me blow this up. So here we have the cell wall in light pink. And as you can see, the cell wall goes around the endoflagella. All right, let's go to the video. There we go. Start that over. Come on. You can see how flexible, there we go. You can see how flexible the spirochete is. That actually changed direction just by bending. And now it moves this way and then that way. I think this shows another one. So what this does is to move this way, it just rotates one way in a corkscrew-like movement. And then to reverse direction, it just rotates the other way. What is unusual is this spirochete, which is only seen in spirochetes, where it can actually bend on itself and then it changed direction. Instead of going north and south, it's going east and west. All right, any question about the endoflagella? I don't see why I'm there. A reminder that the endoflagella do not beat like a whip the way a flagella normally does. When it moves, it causes the cell to move in this corkscrew-like movement. The fimbriae are like hairs on a bacteria or archaea, and they're shorter and straighter and thinner than flagella. They're used for attachment to surfaces. They're not used for swimming. They are kind of important because usually the human pathogens, the part of the cell that will attach to the human receptor, will be on the fimbriae. The pili, also called the sex pili, is longer than fimbriae. In fact, we can see it here. Let me see if I can blow this up. So here's the sex pilus or sex pili, and it's pili if it's plural. If there's one, it's pilus. It's longer than the fimbriae, which this cell actually has several fimbriae. Longer ones here and shorter ones there. The sex pilus is hollow and can allow DNA to move from this cell, which has the sex pilus, into this cell, which is lacking a sex pilus. And we'll talk a little bit more about that when we discuss genetics in chapter 8. So the purpose of the sex pilus is to transfer DNA from this cell into that cell. All right, let's talk a little bit about the cell wall. The prokaryotic cell wall is a complex, semi-rigid structure responsible for the cell shape. In fact, if the cell has a cockeye shape, or a bacillus shape, or even a spiral shape, the reason why they have this shape is because of the cell wall. And that's true also for the weird shapes like the archaea, which is a rectangle. The cell wall is in the shape of a rectangle, giving the cell its shape. The cell wall protects the cell and prevents osmotic lysis. How it does that is if the cell is put in an environment where there's more water outside the cell than inside the cell, water will move into the cell. That will cause the cell membrane to expand, and water will continue moving into the cell until the water is equal inside and outside the cell, or until the water moves into the cell membrane has expanded so much that it bursts. and that will kill the cell. And if you put red blood cells into a beaker of water, water will move into the cell until the cells burst, and then all the blood will be gone. The red color will be gone because the cells have lysed. And that happens because red blood cells do not have a cell wall. But if the cell, like a bacteria, has a cell wall, water will move into the cell membrane will expand up until it reaches the cell wall. Once it reaches the cell wall prevents the cell membrane from expanding any further. If the cell membrane cannot expand any further, water cannot move into the cell anymore because there's no room for it. And that will protect the cell from bursting its cell membrane, because the cell wall prevents the cell membrane from expanding any further. So the cell wall protects the cell and prevents osmotic lysis. The cell wall is located outside of the cell membrane, and it is considered the last part of a cell. Anything outside the cell wall is considered external to the cell. It's kind of interesting that the cell wall is considered the last part of the cell, because the cell wall does not regulate what comes into and outside the cell. That is regulated by the cell membrane. And the cell membrane is actually internal to the cell wall. But the cell wall is considered the last part of the cell. Any question about that? All right, the cell wall can contribute to the pathogenicity of some species, meaning the cell wall can make you sicker. An example of that would be, let's see, 0157H, I think it's 111. This is antigens on E. coli, which is a human pathogen causing gastroenteritis, normally E. coli, which I don't think it has any specific antigens, does not make people sick, does not cause gastroenteritis, but there are some strains that do. And those strains that do have molecules in their cell wall that give the pathogenicity to those strains of E. coli. And then there are species like Mycobacterium, where the cell wall also contributes to the pathogenicity of the species. Any question about any of that? We'll talk about the Mycobacterium a little later in the year. The prokaryotic cell wall is mostly made up of one molecule, a peptidoglycan. And I'm talking about I shouldn't say the prokaryote cell wall, I should say the bacterial cell wall is mostly made up of peptidoglycan. It is the main molecule in the cell wall of all bacteria that have a cell wall. Any question about any of that? We'll talk more about peptidoglycan later. I guess I'm going to talk about it now. We'll talk more about it in a later lesson, but we're going to talk about it now also. Peptidoglycan is a polymer of a repeating disaccharide. Let me blow this up. So the repeating disaccharide that makes up peptidoglycan is NAG and NAM. NAG is a sugar, and so is NAM. And NAG stands for N-acetylglucosamine. NAM stands for N-acetylmuramic acid. And my guess is these names mean as much to you as NAG and NAM. And so you can learn them by either the long name or by the abbreviation. NAG and NAM, just repeat NAM, NAG, NAM, NAG, NAM, NAG in a row. And then you have repeating rows next to this row. And then you have planes of rows underneath and further planes underneath that. Any question about any of that? All right, in the NAG and the NAM disaccharide, repeating disaccharides, they are linked together by short polypeptides. So right here, come on, mouse, this row is hooked together to that row, by this peptide. And then this peptide here hooks this peptide together with that peptide. And so this peptide literally links this row to that row. And that holds the peptidoglycan together, the peptides. The NAG and NAM are, of course, held together by a covalent chemical bond linking the NAM to the NAG and the NAG to the NAM, etc., making the row. But the short peptides hold on to the NAG and NAM, like here to here, and then there to there. And that actually gives peptidoglycan its name. The glycan means sugar, and NAG and NAM are sugars, the disaccharide. And then the peptides hold the NAG and the NAM together. So that's why it's called peptidoglycan. The peptidoglycan held together surrounds the cell wall. And as you can see, there are many layers of peptidoglycan. There's 3 shown here. When we look at it this way, you're only seeing 1 layer, but realize it's many layers of peptidoglycan. Penicillin actually works by preventing the peptides from holding the cell wall together. Like penicillin, before this peptide is put down, when the cell is making its cell wall, it will make the NAG and NAM first, and then it'll start making the peptides. And before this peptide gets put on, and that has to go on after these are made, a penicillin can get in here, and then it'll block this peptide from forming. And when that happens, there will be a hole, a very small hole, but there will be a hole right here where the cell wall isn't held together by the peptide. And then when water comes into the cell membrane can expand out of this hole. And when more water moves into the cell membrane can expand. Come on, mouse. For some reason, there it is. Can expand further. And what will happen is this cell membrane will expand, keep on expanding until it bursts. Sort of like blowing up a balloon in a box. And you have a hole in the box. The balloon will start protruding outside the hole. And if you keep blowing on the balloon, it'll keep protruding until the balloon will burst from having too much air and expanding too much out of the hole. Any question about any of that? All right, let's take a look at once again at the bacterial cell wall. There are 2 types. The gram-positive cell, shown here, where we have a very thick purple layer. That's the peptidoglycan layer of a gram-positive cell. The yellow layer is the cell membrane, and that's a lipid bilayer, mostly composed of phospholipid. It does have other molecules in it, but the main molecule in the cell membrane is phospholipid. In the cell wall, the main molecule is peptidoglycan. and it'll just be layer upon layer of peptidoglycan. Note that here, the cell wall, they're only showing 3 layers. In reality, the gram-positive cell wall will have at least 20 layers of peptidoglycan stacked one after the other, meaning one on top of the other. And it could be hundreds of layers of peptidoglycan stacked one on top of the other. The point is the cell wall is thick, has many layers of peptidoglycan. And I don't think we can see that here. Let me blow that up. No, you can't see it here. There are other molecules in the cell wall, the main one being teichoic acid. It runs through the peptidoglycan and helps hold the peptidoglycan together. There's also lipoteichoic acid, which is teichoic acid with a lipid on it, and the lipid portion will anchor itself into the cell membrane, which is a lipid. The teichoic acid, I don't know if it's important or useful. It is useful for ions to move into the cell. The ions will tend to move down the teichoic acid. However, let me state that for the ions to move through the cell wall, they do not need the teichoic acid. And that is anything that can dissolve in water, which includes ions, can move through the cell wall. The cell wall does not prevent the flow of anything that can dissolve in water. Any questions about that? So water can get right through the cell membrane, and anything dissolved in the water can too. Excuse me, not the cell membrane, the cell wall. Water can get right through the cell wall, and anything dissolved in the cell wall can get through the cell wall also. There can be polysaccharides in the cell wall, which can provide antigenic variation. Antigens are something our immune response will respond to. And usually the antigen is a molecule which is the outermost molecule of the cell. So our immune system usually responds to an outermost molecule, and most of the outermost molecules in a bacteria are in the cell wall. Any question about any of that? All right, the gram-positive cell wall differs from the gram-negative cell wall. Not that they don't have peptidoglycan. The gram-negative cell does have peptidoglycan, but it's a thinner layer of peptidoglycan than the gram-positive cell. You'll note that it is a layer, though. The artist is only showing it, the gram-negative, as one layer of peptidoglycan. In reality, in gram-negative cells, it's 3 layers of peptidoglycan to about 20. So any place between 3 layers of peptidoglycan to 20. The point is the gram-negative has a thinner layer of peptidoglycan than the gram-positive cells. The gram-negative cell also has a yellow area outside the peptidoglycan, and this is called the outer membrane of the cell wall. So it is a part of the cell wall. Well, we call it an outer membrane of the cell wall, and it is a lipid bilayer. So there's the bilayer of the cell membrane, and its main component is phospholipid and it's a bilayer of the phospholipids. Here we can see the outer membrane of the cell wall. Let me blow that up. It does contain phospholipid, shown here and there. In fact, the phospholipid is the most common molecule in the outer membrane of the cell wall. But it does have another lipid in this layer, not in that layer. This layer is very similar to the cell membrane, the bottom layer of the outer membrane. But the top layer of the outer membrane contains another lipid, lipid A. Lipid A is attached to an O polysaccharide, that's the hair shown here. And so this molecule together is called a lipopolysaccharide. The lipid A is the important part because lipid A is an endotoxin. Toxins are something that is toxic to us, meaning poisonous to us, and lipid A is a toxin. That means all gram-negative cells that have a cell wall have a toxin. Any question about any of that? All right. There are other molecules found in the cell membrane of a not cell membrane, the cell wall of a gram-negative cell. The main one is the porin protein, but there are lipoproteins. The lipoproteins run through the outer membrane and may run through the peptidoglycan and then attach to the cell membrane. The porin protein is probably the most important protein in the outer membrane. The porin protein is a channel protein allowing certain molecules into or through the outer membrane as well as out of the outer membrane. Any molecules that the cell would want, such as glucose, fructose, any amino acid, any fatty acid for that matter, they are allowed through the porin protein. And that's how those molecules get through the outer membrane of the cell wall. Any question about that? Now, for the molecule getting through the cell membrane, it doesn't come through a porin protein, which allows many different molecules to cross, like glucose and fructose and tyrosine, the amino acid tyrosine, the amino acid phenylalanine. They all can come through the outer membrane through the porin protein. For each of those molecules, we have one transport protein in the cell membrane, and that one transport protein can only move molecules of one type, meaning you have a different transport protein that can move fructose across the cell membrane, and you have a different transport protein that can move glucose. The one that can move glucose cannot transport fructose, and vice versa. So in the cell membrane, we have transport proteins that generally only transport one molecule across the cell membrane. But in the outer membrane of the cell wall, we have the porin protein that allows many different molecules to cross. Any question about any of that? I think I've talked about everything here. So in the gram-negative outer membrane, we have lipopolysaccharides. That's where lipid A is. the endotoxin. We have lipoproteins and phospholipids. I'm not going to quiz you on the periplasm, but that's the space between the outer membrane and the plasma membrane. This is the space where the peptidoglycan is found. I thought there it is there. So the space in between here and there, that's where the peptidoglycan is. So the periplasm is part of the cell wall. The gram-negative outer membrane can protect the cell from complement proteins. That is a protein that our body makes to help us defend the body from infections. It also, the outer membrane can help us from the complement protein. And we'll talk, can we talk about that? It's a protein that also helps the body's immune system from a foreign invader. And then the outer membrane can also protect the cell from antibiotics. As I mentioned earlier, penicillin G cannot get through the outer membrane of a gram-negative cell. Now, why did I go back to there? I just ended the show. There we go. This is what I wanted to show. The reason why penicillin G cannot harm gram-negative cells is they get in the cell wall and then prevent the link, the cross links of peptidoglycan from hooking the NAG and NAM rows together. So hooking the peptidoglycan together. Well, penicillin G cannot come through the porin protein. And so the outer membrane protects the gram-negative cell from penicillin G. Now, there are other antibiotics which can get through the porin protein, and that includes ampicillin, which is, I think, the third member of the penicillin family to be discovered, as well as any of the modern members of the penicillin family, like carboxycillin. The point is the porin protein can reject some molecules and then allows other molecules to enter the outer membrane. There can be an antigen in the outer membrane of the cell wall. And I did mention one E. coli one. Well, here's another E. coli strain, 015787, another human pathogen. This strain of E. coli causes gastroenteritis. And it's because of this O polysaccharide. The most important thing about the gram-negative outer membrane of the cell wall is it does contain lipid A, which is an endotoxin. So when we look at the gram-positive cell walls and compare them to the gram-negative cell wall, you can see that there's a thick peptidoglycan in the gram-positive cells and a thinner peptidoglycan in the gram-negative cells. There's many layers of peptidoglycan here, 20 to 100 or more layers. And here there's a few till the upper size would be about 20 layers of peptidoglycan. So thin peptidoglycan. The gram-positive cell has a rigid structure from the cell wall. The gram-negative is not as rigid. The teichoic acid is a protein found in gram-positive cell walls. Teichoic acid is absent in gram-negative cells. Any question about that? All right, not hearing anything. There is no outer membrane in the gram-positive cell wall. There is an outer membrane in the gram-negative cells, and that outer membrane contains lipid A and endotoxin. Any questions about any of that? All right. The gram stain mechanism of the cell wall, the crystal violet iodine complex forms in the cell when you add iodine to the cells that are been stained with crystal violet. In the gram-positive cells, the crystal violet iodine crystals do not leave the thick peptidoglycan. At least if you're doing the gram stain correctly. If in the gram-negative cells, the alcohol dissolves the outer membrane. The outer membrane is mostly lipid, so alcohol will dissolve it. And then the crystal violet iodine washes out of the thinner layer of peptidoglycan. So the cells would be clear, but we then stain with safranin, and that stains the gram-negative cells pink. Any question about any of that? All right, this table is showing you a comparison of the gram-positive cell and the gram-negative cell. The gram reaction, the gram-positive cells retain the crystal violet. The gram-negative cells are decolorized and lose the crystal violet. And it's because the gram-positive cells have a thick peptidoglycan layer. The gram-negative cells have a thin peptidoglycan layer, which allows the crystal violet iodine complex to leave the cells in the decolorizing step of the gram stain. Teichoic acid is present in the gram-positive cells. Teichoic acid is absent in gram-negative cells. The periplasmic space is absent in gram-positive cells, present in gram-negative cells. The outer membrane is absent in gram-positive cells, is present in gram-negative cells. And obviously the lipopolysaccharides in the outer membrane, so it's absent or none in the gram-positive cells. I'm slightly making that an oversimplification, but you find it at high concentration in the gram-negative cells. Any questions about that table? All right, so I've talked about the cell walls of bacteria. Let me state that I'm talking about the cell walls of normal bacteria, meaning most of the bacteria. There are some cells which have atypical cell walls. Let me talk about the archaea first. The archaea are not bacteria. They're in a different domain. And so it's not surprising that the cell walls of the archaea differ from the cell walls of the bacteria. Any question about that? The cell walls of archaea do not have peptidoglycan. They have pseudomurine, which is another disaccharide. And some of the archaea lack cell walls. Of course, some of the bacteria lack cell walls also. But if they have a cell wall, which most archaea do, then it's not made of peptidoglycan, it's made of pseudomurine. And I won't quiz you on that. You should just know the archaea bacteria cell walls differ from the bacterial cell walls. And then there are two families of bacteria that have atypical cell walls. First, there's the mycoplasmas, and they lack a cell wall. they do have a plasma membrane, obviously. And curiously, they have sterols in their plasma membrane to give the cell increased strength and support. And that's curious because animal cells lack a cell wall, and we have a sterol that we call cholesterol, which is found in our plasma membrane, and cholesterol gives the cell strength and support, or the cell membrane, I should say, strength and support. And the mycoplasmas have something similar. It's just they don't have cholesterol. It's another sterol. Any question about the mycoplasmas? If you don't ask a question, I'm going to ask a question. How do you think the mycoplasma stain and the gram stain? Are they gram positive, gram negative, or something else? The mycoplasmas. You want to have a guess? Gram positive, gram negative, or something else? Come on, somebody's got to guess. Well, what's the gram stain? What's the crystal violet stain? All right, nobody's answering. Crystal violet stains the peptidoglycan of the cell wall. The mycoplasmas lack a cell wall, so they have no peptidoglycan. So they're not going to retain the crystal violet iodine complex, so they will stain gram negatively. The other atypical family of bacteria are the family Mycobacterium. They have a lot of mycolic acid in their cell wall, and that gives them acid-fast staining ability. Let me end that. I don't think our next slide has it, so I need to go online and show you a picture. If I can get it. There we go. I think this will work. Just I'd like a key to go with it, and I don't think that one has a key. Oh, here we go. This is the cell wall of the Mycobacterium. You'll note that it does have peptidoglycan, which is this layer right here. Beta-lactams. I'm not seeing it in the key here. MA, is that mycolic acid? Yep. P, peptidoglycan. I don't see a P on that picture. There's PG. That is the peptidoglycan layer. And then there are other molecules in the Mycobacterium. Come on. Oops, not what I wanted. I don't see an MA in this either, but there it is there. There's the mycolic acid. So the mycolic acid is a very large amount of the cell wall in the Mycobacterium. Mycolic acid is a lipid, and that gives different properties to these cells. For one thing, water cannot easily enter the cell or leave the cell. And that means anything that's water soluble will have a hard time getting into the cell. And so the mycobacteria are resistant to many antibiotics like penicillin G, which is soluble in water. It won't get through the mycolic acid. The point is that these cells are gram-positive, but they're different because of all this mycolic acid, and the acid fast stain can get into the lipid and then stain the cells. And these give these cells an acid fast staining property. So they stain as an acid fast positive cell, and then a cell that lacks mycolic acid, the acid fast stain will wash out of it. Yeah, let's close this down now. Don't need that. Well, I guess I could take that for the lab, but there is no lab today, so let's go back. We'll talk a little bit more about the Mycobacterium and the acid fast staining ability, but for now, all you need to know is the Mycobacterium and the Mycoplasma have atypical cell walls for the bacteria. The mycoplasmas lack a cell wall. The Mycobacterium have a cell wall where peptidoglycan is not the main molecule. Mycolic acid is the main molecule. And mycolic acid gives different properties to the Mycobacterium. Any question about any of that? All right. Come on. There we go. You can damage the prokaryotic cells. You don't need to know the names here, but you can damage the prokaryotic cell wall by treating the cells with lysozyme, an enzyme that digests the peptidoglycan. You can also damage the cell wall by exposing the cells to a member of the penicillin family. I'm only going to talk about penicillin G, but all the members of the penicillin family work in the same manner, and that is they inhibit the peptide linkage in the cell wall between the peptidoglycan or the glycan rows. Any question about any of that? All right, the gram-negative bacteria are not as susceptible as the gram-positives to penicillin G. And it's because of the outer membrane of the gram-negative cell that penicillin G cannot get in and harm the cell. So it's the outer membrane that doesn't allow penicillin G to get into the peptidoglycan and then do its damage. That doesn't mean that there aren't other members of the penicillin family that can damage gram-negatives, like ampicillin and carboxycillin. They can damage both gram-positives and gram-negatives. And there are other antibiotics that do not damage the gram-negatives. That doesn't mean that the gram-negatives are not susceptible to antibiotics. You just need to use different antibiotics. Like streptomycin is an antibiotic that is lipid soluble. So this antibiotic can get right through the outer membrane of the cell wall and then damage the cell. Another is polymyxin B, which only works on gram-negative cells. It does not work on gram-positive cells. What it does is it puts a hole in either the outer membrane of the gram-negative cell or in the cell membrane, also of the gram-negative cell. And so these antibiotics can kill gram-negative cells. All right, let's talk a little bit more about the plasma membrane. It is a phospholipid bilayer where the phospholipids come together and then come together to form a lipid bilayer shown here. Let me blow this up. So they form a bilayer here, coming together one after the other, where the fatty acid tails come on the inside because they like to be around each other because they're lipid loving or hydrophobic. And then the heads of the phospholipid face the water inside the cell, the cytoplasm, which is mostly water. And then outside the cell is also mostly water. This is particularly apparent if you're talking about a red blood cell facing the blood. The blood is mostly water. But that's true of any cell, even a bone cell, because the majority of our tissue is mostly water. So the heads face the mostly water outside the cell. You can have proteins in the cell membrane or the plasma membrane. There are three types of proteins: peripheral proteins, integral proteins, and transmembrane proteins. A peripheral protein is a protein in the cell membrane where most of the protein is on one side or the other of the membrane. So that's a peripheral protein. An integral protein or a transmembrane protein is a protein that spans the membrane. So that would be a protein that spans the membrane, and so is that one. This one looks like it's either a channel protein, allowing a molecule to either leave or enter the cell, or it could be a transport protein, which actively pumps a molecule into the cell. And there are a few active transport proteins that pump a molecule out of the cell. I just can't name any off the top of my head, but glucose transport protein or lactose transport protein is an active transport protein which will pump a molecule from outside the cell into the cell. Any question about any of that? Oops, let's move on. I think I can finish this slide in time. The membrane is actually a fluid. It's kind of hard to realize that my hand is held together only by cell membranes, but that hand is held together by fluids. The cell membrane is a fluid, and we use the mosaic fluid model or fluid mosaic model to explain the cell membrane. That is, the membrane is a mosaic of phospholipids and proteins and a few carbohydrates as well. The membrane is as fluid as olive oil. As you know, olive oil is pretty fluid at room temperature. But if you were to put it in the refrigerator, olive oil would solidify, or at least a portion of it. The proteins can move about in the membrane. And those of you who have had AMP, you already know that when a protein binds to a receptor, those receptors usually move together before they come into the cell and then trigger their signal. And that's because most of the proteins can move about to function. there are a few proteins that do not move in the membrane. And those would be proteins which are anchored either by the extracellular matrix or the intracellular matrix. So if this protein were bound to an intracellular matrix protein, that protein is not going to move. If this protein is bound to a extracellular matrix protein, let's say there's like a bone cell here and it's anchored to the bone, that's the extracellular matrix, then this protein will not move in the membrane. But all the other proteins are free to move in the membrane. The phospholipids also can both rotate around as well as move laterally from one place to another in the membrane. Any question about any of this? If not, I'm going to end it here. That's exactly 6:20. And if you have a question, I will be in the lab for the first 15 minutes of the lab. But if you don't have a question, you don't have to show up. Okay? All right. Any questions before I log out? All right. Goodbye. I'll see you next.